Hello, everyone. Since today we have three excellent group of speakers, let's start on time. Our first speaker is Hubert Huang. He is a research scientist at the Broad Institute under the Sellers Lab. He obtained his bachelor degree in chemistry from Duke University and his PhD in chemical biology from Harvard University under the guidance of Professor Nathaniel Gray and Professor Jane Zhao, and where he developed the kinase inhibitors and degraders as a cancer therapeutics. Hubert, we are very much looking forward to your seminar.
Thank you. Thank you, Mikolai, for the introduction, and I want to thank the organizers for the opportunity to present here today. It's really my honor to share the stage with the two labs that developed BioE3 and UBPod. It's honestly quite validating to find us arriving at a very similar strategy to identify E3 ubiquitin ligase substrates, and I hope our presentations today will be a great resource for the field of targeted protein degradation as well as E3 ligase biology. So one of the kind of important question that the field of targeted protein degradation is trying to answer is how is the molecular characteristics that determine the efficiency of substrate degradation. And while many efforts have been spent on trying to understand how protein degraders bind or form ternary complex formation with the substrates and E3 ligase, as well as using total proteomics to study protein degradation. However, protein ubiquitylation has mostly been kind of skipped or ignored, perhaps because it is still quite a finicky post-translational modification to study, and also perhaps most of the therapeutic hypothesis focus on degradation. But we think by skipping the understanding of protein ubiquitylation, we could be missing very important insights. And furthermore, for the over 600 E3 ubiquitin ligases encoded in the human genome, we still have very limited understandings of their functions and substrates. And we think by understanding these questions better, we could potentially develop better ligands to target these ligases, as is the case for VHL ligands, and also to understand the impact for targeting these ligases, either as for therapeutic impact or for potential off-target effects. And for these reasons, our goal is to develop a method that can detect the ubiquitinated substrates of E3 ubiquitin ligases. And we were also very inspired by all the recent progresses with the field of proximity labeling. And so I set out to combine the two aspects this led to the development of ESTUB, which stands for E3 substrate tagging by ubiquitin biotinylation. It consists of two components. One is a BRA tagged E3 ubiquitin ligase, and the other is a BAP tagged ubiquitin or biotin acceptor peptide labeled ubiquitin. So this BRA enzyme is the wild type version. So that's different from the bioID that promiscuously biotinylate proteins in proximity. This version of BRA actually have no substrates inside human cells, but would instead only specifically biotinylate peptides. Here we call BAP. And so we envision that when you co-express these two components inside the cells, we can biotinylate ubiquitinated substrates that are interacting with the E3 ligase of interest. And this will, the strong interactions between stripavidin and biotin would enable a stripavidin-based enrichment followed by identification of putative substrates using Western blots or mass spectrometry. So to test if this method would work, we set out to see if we can detect ubiquitinated substrates of known E3 substrate interactions. So here, coming from the field of targeted protein degradation, we utilize the system of CCA5 induced ubiquitylation of GSPT1 by Sagar Bhogaraju, we expressed cereblon-BRA and AV tag ubiquitin, which is a form of BAT tag ubiquitin, in 293T cells. And we treated cells with proteasome inhibitor as well as CCAA5. And when we analyzed the biotinylated proteins enriched by strip-avidin, we found GSPT1 to be highly enriched and ubiquitolated upon the addition of CCAA5. However, we also noticed pervasive background biotinylation, and we reasoned that this is because AV tag is such a good substrate of BRA that it readily gets biotinylated, and we're not getting much of proximity dependency. And inspired by prior work from Alice Ting's group, we explored AV tag derivatives that are shorter in length and have less of an affinity toward BRA, and ultimately arrived at A3 ubiquitin, which preserved GSPT1 enrichment while showed a greatly reduced background. And we think this was the ideal version to move forward with mass spectrometry studies. So when we compared these two samples analyzed by mass spectrometry, and this is done in close collaboration with Eric Fisher Lab, and specifically Ryan and Catherine have been tremendous support for these efforts. And so when we compared these data in the volcano plot, we could see GSPT1 and 2, the known substrates induced by CCA5, to be greatly enriched, and it's highly significant. And we also found ETF1 and HBS1L, which have been previously reported and are on mechanism collateral or paralog proteins that will be ubiquitolated and in certain cases degraded by CCA5. But interestingly, we found vinculin, a commonly loading control, to also be induced ubiquitolated. And we were quite surprised, but when we validated our results using Western blot, indeed we saw induced ubiquilation of vinculin upon addition of CCA5. And this level of ubiquilation actually does not lead to its degradation, which is likely why it has been missed previously. And while so far we still don't really understand the biological impact of this type of induced ubiquilation, this showcased that ESTEP really can provide a more direct and complete view of the impacts of remodeling the activities of an E3 ligase by directly interrogating ubiquitylation activities. And next, we wanted to see if ESTEP can apply beyond just cereblon. So here we, and also wanted to explore the substrate scopes of the method. So here we utilized 4 PROTACs that could either recruit cereblon or VHL to the proximity of BET bromodomain proteins or the kinase PTK2. And when evaluated in the specific models, either overexpressing cereblon-BRA or VHL-BRA, we could always identify the expected hits as the most significant ubiquitolated proteins. So this really demonstrated the robustness of our method. And next, I wanted to explore this question of, like, do we really learn new information by studying induced ubiquitylation? And is it fair to make the assumption that if something is degraded, then they're being ubiquitolated? So in order to ask these questions at scale, we turn to multi-kinase degraders, which are PROTAC molecules that can bind to many kinases. And this is an efficient way to assess the ubiquitylation and degradation of many proteins with one probe molecule at one time. And so we use this compound called SK391. And at the bottom, you see the ESTEP profile of this compound. There are many proteins to be induced ubiquitolated. And when you read the gene names, you could find that the majority of them are kinases or kinase-associated proteins. And that goes on to show how specific and robust the method is. So when we compare the ESTEP profile of SK391 at 1 hour treatment to a 5-hour treatment of SK391, but evaluated using total proteomics. Total proteomics being on the x-axis, highly degraded proteins are on the left-hand side, and highly biotinylated proteins, highly ubiquilated proteins evaluated by E-stub are on the y-axis and moving toward the top. We found that in the yellow zone, many proteins indeed are both degraded and ubiquilated. However, we also found many proteins to be showing signs of significant ubiquilation without significant degradation. And we think we haven't really done too much in-depth analysis of select hits in this zone, but we think this mostly represents subproteolytic events. So they're definitely showing significant ubiquilation. Perhaps the chain lengths are not long enough to lead to efficient degradation. But this goes on to show how powerful the method is, because these hits would have been buried within the noises, and you wouldn't have been able to confidently call them to be degraded using total proteomics. But we're definitely seeing signs of induced proximity as well as ubiquilation. And if these are therapeutic targets that are of interest, then medicinal chemists can have more confidence in doing medicinal chemistry to further improve target ubiquilation and degradation. This may also represent off targets of collaterally ubiquulated targets that might be missed using total proteomics, but may have important implications in their certain compounds usage in the clinic. We also found many or several proteins to be degraded, but not significantly ubiquulated. And since most of them are not kinases in this case, we think this likely represents secondary effects to the pleiotropic effects of a multi-kinase inhibition and degradation at the 5-hour time point. And last but not least, there are many proteins that were found by E-stub that wasn't even quantifiable or detected using total proteomics. And again, these are kinases in addition to several zinc finger proteins that have been previously reported to be substrates of IMiD or IMiD-like molecules. And this really goes on to show the power of just doing one single profiling in a single cell line with a multi-kinase degrader, and how much more information we can learn from it. And due to the interest of time, I will skip this story, but this is already published. And I would end my presentation on this slide talking about the direction of using E-stub to identify endogenous substrates that we're really excited about. So here we're demonstrate using VHL as a proof of concept to show that by comparing wild type to mutant VHL, the form that either cannot interact with HIF1 alpha or have lost its ability to assemble into an effective COL2 CRL2 complex, that in using either strategy, you can identify HIF1 alpha as a clear hit, a clear substrate of VHL. And so after me, Rosa and James will talk about bioE3, and then Sagar will talk about UbiPod, and there they would expand it beyond cullinating ligase systems to ring and HEC type ligases as well as non-ubiquitin ligases. So yeah, in conclusion, E-stub is a ubiquitin-specific proximity labeling method that is applicable to different E3 ligases and a broad range of substrates. And since it directly interrogates protein ubiquilation, it provides additional insights to the molecular mechanisms that is complementary to global proteomics. And E-stub may be a general strategy for E3 substrate mapping, which can potentially shed light on the physiological roles for over 600 E3 ubiquitin ligases in the human genome, and even ones beyond ubiquitin ligases to SUMO and other ubiquitin-like proteins. And the relevant plasmids in this study have just became available on Addgene. And yeah, I would like to acknowledge the guidance of people on my team, Ryan, Shuyao, Nate, and James. And also really appreciate the mass spectrometry support and expertise from Eric Fisher, Ryan, and Catherine. And I would hand this to Rosa and James.
Thank you very much, Hubert. And now, Brian, I will introduce our second speakers for today, and we'll try to take all the questions at the end of free seminar.
Great, thank you. So it's my pleasure to introduce today Rosa Barrio and James D. Sutherland, who are principal investigator and associate PI at CIC bioGUNE in Bilbao. The lab there focuses on ubiquitin-like modifications in development and in disease. They both joined CIC bioGUNE, which is accredited as a Cervero Ochea Center of Excellence and is part of the Basque Research and Technology Alliance, where they study rare diseases such as Townes-Brocks syndrome and develop biotin-based proteomics tools for the cure of ubiquitin-like pathways. So with that, I will hand it over to you for your presentation.
Okay, thank you so much for this invitation, and it was a great idea from Hubert to put the three groups together, and we really appreciate it. And so we will try to show you the things that we do using this bio-E3 strategy to for the identification of specific substrates of E3 ligases. So I don't need to talk to this audience how important the ubiquitin proteasome system is to maintain the physiological cellular functions. And because there are mutations in this system that they lead to disease, specifically we are interested in mutations in E3 ligases like UB3A, MIP1, MIP1, etc., that they lead to many rare diseases. Problem is that to study these, they are not known all these possible targets of these E3s, which difficult to make difficult to explain the pathologies. In the lab, in particular, we are interested in the Townes-Brocks syndrome. This is caused by mutations in the transcription factor SAL1, and the patients have ciliopathy-like phenotypes, and even though it's not an E3 ligase, it does cause protein homeostasis defects. So, for the reason that the difficulties to study ubiquitination in the cell, we turn to the Behrends biotin system for doing those studies.
Okay, and as Rosa said, we've been working with the biotin system for studying ubiquitin for quite a while, and Hubert already introduced this idea that BRA is a ligase, that it can put biotin onto a specific tag in the cell. We've used that idea to develop various tools for studying ubiquitin system. You can fish for entire ubiquitomes. You can look for interactors of ubiquitinated proteins using SUMO-ID or UBL-ID. But we wondered whether these tools could help us with the problem at hand. Can we use the system to study targets of specific E3s? And just like the other speakers today, we had the idea that, okay, if we fuse BRA to an E3 of interest, when that E3 is putting a ubiquitin onto a substrate, if that ubiquitin is avi-tagged, then maybe the BRA will drop a biotin on it, and then we can catch it. The problem is, as Hubert said, we have a lack of specificity, and we have been optimizing the system, and basically the first key point is that avi-tag is designed to have very high affinity for BRA, and as soon as it's translated these two proteins, they label each other, and you lose the proximity. So we use a low affinity version of the avi-tag called bioGEF to accomplish this system. Other modifications that we've made is that we culture the cells in biotin-free medium to reduce the background of biotinylation in the cell. And then when we're ready, we pulse the cells with biotin, and this really improves the signal-to-noise ratio. To suppress recycling that might happen of this biotinylated ubiquitin, We use a mutant form that is basically it's not cleaved well by deubiquitinases, so it gets put on, but it doesn't come off very readily. The last point is that we try to control the expression levels in the cell by making an inducible bioGEF ubiquitin non-cleavable cell line.
So as a proof of principle, we started studying RNF4. RNF4 is a ring E3 ligase that is particular in the sense that contains this sumo interacting motif. So it's able to recognize sumo-related proteins and ubiquitinate them and send them to degradation. And it's a ring ligase that is mainly nuclear. So we actually use mutants of both the SIM domains and also the catalytically domain of the enzyme as negative controls. And as you can see, both in the Western blot and in the immunofluorescence, we could see biotinylation of material with a wild-type form that is much more reduced with the delta SIM or the catalytically inactive forms. And we can see both also in the immunofluorescence how these biotinylated forms accumulate in the what could be PML bodies in the nucleus labeled with fluorescence streptavidin. So encouraged by those results, we sent the samples to mass spec, and we saw in fact that we found many components of the PML nuclear bodies, which made sense, and other RNF4 targets. Also, we saw that we could find sumo among the ubiquitinated material, indicating that the main target of RNF4 are sumo-related proteins. In addition, we could see that when we add ATO, the arsenic trioxide treatment, we promote the ubiquitination of PML, which happens in the wild type, but not in the mutant forms, which also was very encouraging for us to see that the system is actually chemically responsive. Another E3 ligase of our interest was MINBON or MIP1. This is also a ring ligase that localizes in the centriolar satellites and in the centrosome. And that also we were able to make a mutant as a negative control. And we could see that the wild type accumulates biotinylated material in comparison with the mutant, and that this material actually accumulates also in the pericentriolar material as well as the E3. By mass spec, we found centrosomal proteins. We were able to validate some of them with the wild type form, but not with the mutant. And we were puzzled because we found also many members of the autophagy receptors ubiquitinated with MINBON, and until we found out that in fact MINBON was previously associated with autophagy. So it seems like a very specific system to able to recognize the targets of these two different ring ligases.
So we were content that the system was working for ring ligases, and we thought maybe we'll try a different kind of ligase. HEC ligases, as you know, are different, that the charged E2 transfers ubiquitin to the E3 ligase, to the HEC domain, before it transfers to substrates. And we took NED4 as a candidate HEC ligase, and we collaborated with Simona Polo's lab, who's an expert in NED4, and we discovered that NED4 is auto-inhibited, and this kind of gave us some problems. But Simona knew of a mutant that was it had several mutations that led to an open form, which was activated. So we used this open form. We also used the catalytic dead version that mutates in the HEC domain. And we could see that using the wild type form, we get good labeling, but not with the catalytic dead. And that's also the same for the immunofluorescence. When we did the mass spec, we were happy to see that we got several known NED4 substrates, such as WBP2, EPS15. Oh, sorry. EPS15. But also we found several components of the CCT complex. This is a chaperonin complex that was previously not described to be linked to NED4 activity. Okay, so we have also validated bioE3 for some other ring ligases linked to different organelles. one orphan ligase that nothing was known, that we found some new substrates. And we are currently doing some more work with NED4-like, which is linked to a rare disease. And also we wondered whether bioE3 would work for other ubiquitin-likes. So we work on SUMO, and so we tried the system using POS1, POS4. These are two SUMO E3 ligases, and happily we saw that the wild-type forms were able to incorporate, but the catalytic dead versions did not. We still haven't done mass spec for this, but it looks promising.
So in addition of testing the system with other ubiquitin-likes, we decided also to test the system with complex E3 ligases, in the case of the cooling ligases. And from now on, what we'll show you is unpublished results. And we turned to cereblon. We were interested because, as I mentioned at the beginning, we work on a SALF family mutant causing the Townsburg syndrome. And it is known that SALF4, one member of the family, is a neo-substrate of cereblon upon treatment with molecular glues, with IMiDs. So we decided then to test cereblon, and we use SALF4 YFP as a proof of concept in presence of POMA, and we were happy to see that we could detect the biotinylation of SALF4 that didn't happen with a mutant that is insensitive to the pomalidomide treatment. We saw this also by immunofluorescence. You can see better here that actually with pomalidomide treatment, we find the biotinylation in the nuclear bodies where SALF4 normally resides.
And so we processed the samples from cereblon bioE3 for mass spec, and we apologize because the volcanoes are quite empty there, but we were happy to see that one of the endogenous substrates that we got was glutamine synthase, which was previously described to be an endogenous substrate of cereblon. And we've done a data set with pomalidomide, and we think we've identified some additional potential neo-substrates of cereblon, and we're in the process of following some of those up. And the other striking thing that we saw was that when you treat with pomalidomide, some of the endogenous substrates, they actually get reduced. And so we think that in addition to molecular glues serving as a way to target new things, they might also interfere somehow with the targeting of endogenous substrates.
So in conclusion, we can say that bioE3 is a B-ray-based proximity ubiquitination assay that is able to identify the specific substrates for E3 ligases. It can be used for multiple classes of E3 ligases, also multiple ubiquitin-likes and different cell types, and also is compatible with that treatments and stimuli to detect changes in the E3 specific ubiquitin. In progress, we have to try bioE3 in different cell lines to see a specificity of different ligases depending on the context. We want to scale up to more ligases, especially those involved in rare diseases, and to optimize the protocol.
And that's all we wanted to tell you today. I want to point out the website for the lab. Because these experiments are sometimes difficult to set up, we are actually trying to set up a platform for doing these kind of things. And so please feel free to check out the website and feel free to contact us if you're interested in any of those services or techniques. And like Hubert said, we've deposited the reagents at Adgene, so just go over there and pick them up if you want to try.
And of course, we have to acknowledge many people. First of all the lab components for the contributions. In this case, specifically, the main players of this project were Ori Barroso Gomila, who is not longer in the lab, is doing his postdoc somewhere else, and Laura Merino, who is doing the PhD in the lab. And we are very grateful to our collaborators, especially the proteomics facility in CIC bioGUNE in Bilbao, Hugo Mayor at the university in Bilbao as well. Special thanks to the group of Simona Polo in IFON, Milan, Italy, and also the group of Alfred Vertegal in Leiden. So with that, we also thank the audience, and we pass to the next speaker.
Thank you very much.
Thank you.
Great. Thank you, Jim and Rosa, for such a great talk. As mentioned after Hubert's talk, if you could just add your questions into the Q&A, and we'll address all three talks at the end. And with that, I will hand it over to Sam.
Hello, everyone. Today is my pleasure to introduce Dr. Sagar Bhogaraju. Sagar completed his doctorate studies at the Max Planck Institute of Biochemistry and his postdoctoral work at the University. He's now group leader at the European Molecular Biology Laboratory in Grenoble, France, where his lab focuses on the study of ubiquitin signaling in disease and physiology. With that, I'd just like to hand it off to Sagar.
Okay, thank you, Sam. I'm not able to start my video. Is there something that I can do about it, or just start? Okay. Cool, thanks. Okay, firstly, I would like to thank the organizers for inviting me, and it's an excellent idea to put three of us together. I think we can discuss the differences at the later part in the discussion. So I'm a group leader at European Molecular Biology Laboratory. I come from Grenoble, France. And unfortunately, because of the two talks earlier, you will hear some repetitive things, so I apologize for that already. Just to many people might not know where Grenoble is, France is, so I'm going to tell you. So we are located in this small little building here. This is Grenoble. It's typically known for structural biology research. That's why you see a synchrotron here. We also have a nuclear reactor right next to us, which is used for neutron-based structural biology research.
Sagar, excuse me, but I don't know if you're in full screen mode.
Sorry, I am in full screen mode.
You are okay, because we see something different here.
All right.
Yeah, we see only your other screen probably.
Okay. Do you see something here or not?
You might need to click at the top of your presentation to use slideshow. Right now, we're seeing just the PowerPoint.
Okay, how about now?
Great.
Yep.
Okay, perfect. Okay, sorry for that. Just wanted to show where we work. Here in this small little building is where our group is located, and typically our group is doing structural biology work, and we are next to ESRF synchrotron and also nuclear reactor, which is useful for neutron-based structural biology research. If you zoom out, we are surrounded by beautiful French Alps, so if you happen to be around, we'll be happy to take you around for a hike. Now towards the science, we are interested in ubiquitin signaling in general. So part of my group works on ubiquitin signaling in Legionella infection. This is a topic that I inherited from my postdoc. Most of my lab also works on actually melanoma-associated antigens, so-called MAGE proteins, and their roles in ubiquitin signaling in humans. And actually, when I started my group, we wanted to identify some substrates of MAGE-ring ligases or other ligases that we work with. And we found ourselves with lack of expertise and resources. So that's when I thought maybe we should do something about it. It was started as a really small project that I was doing myself in the lab. But then a great postdoc joined, Urbi, who's an EMBL postdoctoral fellow. Most of the work I'm going to show you is actually done by her. And then thankfully also Christian and Sophie joined a little bit later and were instrumental in forming the story that I'm going to show you today. So as I told you, we are structural biologists, so I like to stare at structures. So I was staring at these really beautiful structures of intermediate state complex that is formed between ring E3 ligase, in this case RNF4 ring domain. And here is the E2 and ubiquitin. This is a catalytically intermediate state right before ubiquitin is transferred to the substrates. And this is where I saw that the N-terminus of ring domain and the N-terminus of ubiquitin facing towards the same direction in the solvent. And in fact, I looked at many other intermediate structures, and most of them well, expectedly, the catalytic domain of ubiquitin ligase is close to the ubiquitin during transition states. And another interesting thing I looked at is most ring ligases, around 600 of them in humans, have ring domain, either the N-terminus or the C-terminus, so they're easy to tag. And as you know, HEC ligases have HEC domain at the C-terminus, so they're also easy to tag at the catalytic end. And that's where basically my idea originated. So without fusing BRAE to the ligase and accept a peptide to the N-terminus of ubiquitin, and hoped that when they meet at some point during ubiquitination, BRAE can deposit biotin onto ubiquitin, which can then be transferred to the substrate, and substrate can be isolated. We first applied this method to our favorite ubiquitin ligase, RAD18, which is a ligase that is involved in translation DNA synthesis. So when DNA gets damaged, replicative polymerase gets stalled, and these polymerase needs to be replaced with a more accommodative and error-prone polymerase. And this is done by the RAD18 ubiquitin ligase, which ubiquitinates the DNA sliding clamp, in this case PCNA. That ubiquitination allows for the replacement of polymerases. So the PCNA is the main substrate of RAD18, and we were hoping maybe there are other substrates that we can identify using the method. Okay, so we did the typical experiment where we co-express the acceptor peptide tag ubiquitin and the RAD18 tag to BRAE. We expose the cells to UV or not, and then we isolate the biotinylated proteins and identify them using mass spectrometry. And this was our first experiment, and we were actually quite encouraged to see PCNA as one of the main hits identified that is dependent on UV here. We were also encouraged to see MAGE4, which is a known binder of RAD18. It seems to be ubiquitinated when the cells are untreated. Encouraged by this, we wanted to improve on this method. We saw two basic challenges. I think both of them have been highlighted by the speakers earlier. And one is the acceptor peptide sequence itself. It was developed to be highly specific and to have high affinity to BRAE, which is a problem because you don't want to have biotinylation independent of ubiquitylation. That would lead to identification of a lot of false positives. And that's something that we wanted to tackle, and we and the second is the tackling ubiquitin recycling. As you know, there are DAPs in cells, and ubiquitin is constantly being recycled either by proteasome or other DAPs. In this case, something undesirable happens because the substrate that is biotinylated here will lose biotin when the DAPs act on it, and this ubiquitin is then free to go to any other substrate. So we need to address it, or at least reduce it to have a positive effect on the method. So I'm going to show you two more examples of ligases. With each ligase, we learn something new, and then towards the end, I talk about how to apply this. So as already showed by earlier speakers, there was a nice paper in 2008 which described variants of acceptor peptide that are more proximity specific. And we landed on two variants that were described in the paper, and we tested them in the experiments. And as expected, both variants had very low background biotinylation and didn't have significant impact on the experiment itself. So, and we narrowed down on these -2 AP ubiquitin for our further experiments. So when we use now this new acceptor peptide variant in the UV-POD experiments, now we can not only see higher enrichment of PCNA compared to before, but we also identified more relevant candidate substrates here. And on top of that, we next tried, as I explained to you, tried to tackle the ubiquitin recycling problem by adding a pan-DAP inhibitor, PR619, which is able to inhibit 5 different classes of DAPs altogether. We added to cells right when we also treat the cells with UV. And this had a mixed results, because although we enriched the substrates more, we also found more non-specific hits in this experiment. So this needs to be looked at. But in general, we found with all the ligases we have tested so far, adding this inhibitor improves the substrate enrichment and pushes more proteins towards significant levels in the identifications. All right, so then we were specifically interested in this particular substrate, POL-E, because as I showed you earlier, POL-E is a catalytic subunit of this polymerase epsilon, which needs to be replaced by rad-18. So we thought it is possible that rad-18 also ubiquitilates Pol epsilon in addition to PCNA, because they're very close to each other. And to test this, we knocked down rad-18, and in a dose-dependent manner, we saw increase in the levels of POL-E, and also in a UV-dependent manner as well. So this indicates to us that rad-18 might indeed also ubiquitilate Pol epsilon in addition to PCNA. We're investigating this further. What is the role of this? In addition, we also saw an opportunity to use this method to localize ubiquitination in cells directly. So here is an experiment where we localize DNA damage with a marker that is histone H2AX phosphorylated. And then when we use fluorescent streptavidin, we can nicely see co-localization of biotinylation as well as this DNA damage marker when rad-18 UV pod is performed, indicating that it is indeed possible to see where the substrate ubiquitination is happening in cells, although it might not be true for all the ligases, but it's at least true for rad-18. So next, another ubiquitin ligase that we employed UbiPod on is a CHIP. CHIP is a ubiquitin ligase that is involved in protease protein quality control, and it is neuroprotective. It degrades proteins that are misfolded or prone to aggregation, and therefore plays a role in neurological disorders. It's also heavily studied, and this is where we learned something new as well, because our earlier experiments with CHIP were not working, and then we realized why it might be the case by doing some trial and errors. And one of the things we tried was, I mean, if you look at this structure that I showed you earlier as well, and I saw this is a crystal structure, the distance is 21 angstrom. Of course, in solution that may vary, but depending on the ligase as well, and the extra sequences that each ligase has, either to the N-terminus or C-terminus, this distance may vary a bit. And since the structure, catalytic intermediate structure for CHIP was not available, we just thought maybe we play around with the linker between B-Ray and the ligase in this case and try to see if something works. And after a lot of trials, what we found was actually introducing a small flexible linker between the protein and the B-Ray. In this case, only in this case, we saw efficient biotinylation inside the cells. In all other cases, we failed to see that. So it's possible that the degrees of freedom between B-Ray and ligase also play a role in this method. And we went on to apply this method and use mass spectrometry to identify a lot of known substrates, but also quite an interesting candidate, annexin 5A, which we were highly interested in because there was a recent paper in which they compared the total proteomes of iPSC-derived neurons, comparing wild type and knockout versions of these neurons, and they found that several annexins are actually highly enriched in the knockout. And these include also annexin 5. So we wanted to see if annexin 5 is indeed a substrate. So we treated the cells again. We knocked down CHIP in the cells. And again, we saw a dose-dependent increase in the levels of annexin 5A that is also dependent on proteasomal activity. So telling us that annexin 5 is involved in actually cytoskeleton regulation. So there might be other ways that CHIP is contributing to these neurological disorders apart from degrading the aggregated proteins. So finally, yeah, this is the summary. I mean, we believe that UbiPod is also highly customizable, and it may work for a lot of different ligases, but you have better chance of working if you customize it specifically to your case. And it may work better if you customize it, even though it works suboptimally for probably all the cases. And these are the things that probably need some customization. So first of all, where BRA is fused to the ligase. Is it either N-terminus or C-terminus? What is the linker length? These are all the parameters one can play around. And secondly, what is the control in your mass spectrometry experiments? Either are you using a mutant of ubiquitin ligase, which could be a tricky if your ubiquitin ligase is dimerizing with endogenous ubiquitin. So you can also use BRA alone because it doesn't produce a lot of background. As obviously cell line of choice is also a parameter. And we also found that I haven't shown you the exposure length of biotin to the cells also matters a lot and depends on the ligase as well. So with this, I'd like to thank my group and Ubi, especially who performed most of the work, and also Christian, as I mentioned, is crucial to this project as well, and EMBO and French funding. So thank you also for listening. I think now we can answer some questions.